talk today about the work we've been doing in Cuatro Ciénegas, Coahuila, that has to do with what we want to understand, which is how this interplay between the genotype and the phenotype, and how that is actually influencing the whole community and the ecosystem. So I'll give you, a short introduction to the geology of Cuatro Ciénegas and what we've done with the different genomes and the sampling that has become a very interesting model for the lab to work for many years now. And then I'll give you two stories on the things that we found in the lab. So I love to show students particularly this picture because it's sort of like a geography exam. I was like, can you find Coahuila here? And everyone wants, immediately finds Coahuila somehow here because they think this is the Baja California Peninsula, but it's not at all. I mean, we are talking that Coahuila was part of the ocean before. So even before the Gulf of Mexico emerged, it was completely formed. We have that Coahuila was here. It had a beautiful beach. You didn't have to go all the way to Cancun. And then later, this beach just got there in the middle of nowhere. But still, what we have is that the water was kept as fossil water, producing these beautiful ponds. And these beautiful ponds have interesting things like nothing, like you don't see big fish and you don't see big anything because it's very poor in nutrients. The main nutrient that is lacking is phosphorus. It's one of the places in the world with the least phosphorus. And one other interesting thing is that these sort of stones that are actually stromatolites. And stromatolites are really layers of bacteria that are alive in the case of Cuatro Ciénegas. And these bacteria, what happens is that have a surface that is covered with this mucilaginous thing that can, when minerals are precipitated, are sort of like made into stones. So then bacteria migrates to the top, and the top bacteria are usually cyanobacteria that capture the sun, and then you have a full community of what are called microbial mats. And these microbial mats are so interesting because those are the evidence of the presence of microorganisms on Earth. So of course, that's the kind of fossils you would expect for bacteria. You find these stones where you have evidence of layers with different chemical compositions that are the evidence of the different metabolic strategies already present in the Earth. So even before the Earth had this is the atmosphere, like Mexico City now, and that was before how the world was evolving. Before the cyanobacteria invented this oxygenic photosynthesis and changed everything. But even then, what you had that even phosphorus, because of the lack of oxygen, phosphorus was linked with these kinds of links, like phosphorus directly to a carbon, making these phosphonates. And that explains why only bacteria can use phosphonates, because those were the only things living here. Nowadays, most eukaryotes use this other form of phosphorus. So, a long time ago, with a group of students, we went with Valeria Sosa and decided to sample this place, which is the Churince Pond. And in this place, we placed some sticks, and these are our sampling sites. And we keep sampling sites, since that time. We still sample these sites. And the sampling method that we used then was just use a tiny thing, an epitor tube, and the samples are only 50 microliters large, because we wanted to have bacteria in a very small sample, so that we knew that each bacteria we isolated could be forming a small community in which bacteria would influence each other. And so since that time on, we've had these samples. We have about 3,000 bacillus from the sample. So although there are a large number of bacteria in communities, especially in sediment, sediments are the most rich samples in the earth with bacteria. We only work with samples that we heat treat to get bacteria that are part of the bacillus samples. And the things that we're interested in understanding is, what's the interplay between phenotypic plasticity and genomic plasticity. The plasticity at the phenotypic level is usually short term, and we see evidence of that in things that we call acclimation. You can imagine that when we take our soccer team to Brazil to sort of acclimate to the altitude conditions so that they win the games, and then they don't win. And it's not that they are not prepared, it's that they don't have phenotypic plasticity, okay? So we have to choose our, sports teams, that have phenotypic plasticity. I think that the Germans, that's what they do. Now, if you know what genotypic plasticity, then that's long term. That implies a change that is not reversible. It involves evolution, and you have mutation, and you have recombination, and all kinds of rearrangements. So phenotypic plasticity in organisms can manifest in development, inducible defenses, maternal effects, epigenetics, but typically in bacteria we think about environmental induction and acclimation. And then what happens is that plasticity can expose some variation that is cryptic, and you only detect that when the environment changes and then selects precisely for those phenotypes that are better adapted to the changing conditions. And what is interesting about this plasticity is that the phenotype is going to respond to the change in the ecology, but the ecology is going to respond to the phenotypic change. So in that way, from genes to phenotypes, of course, genes are the base of everything, and that allows the phenotypes to be expressed. But then everything is connected. So the phenotypes are connected to populations, to communities, to ecosystems. And it's really complicated to understand this, but my lab is really obstinate, and one day we'll try to figure out if we can understand this full interplay. And one of the things we know is that plasticity also has to do with the historical conditions in a system. So plasticity is going to facilitate the colonization of new environments, and depending on the genetic background, then plasticity is going to be more or less extensive. And then evolution and plasticity are not directions that a bacteria will take. It can take both. It's not something it's going to select from. And then these plastic changes are expressed by the individuals. They have a genetic basis and also can be in the form of adaptation. So how do we evaluate plasticity? We just heard a talk by Enrique in my lab. And usually what you do to evaluate plasticity, you can do a manipulation of the environment under different conditions, like temperature or pH, or you can look for natural populations. You can take plants that grow at different altitudes and then measure the size of the leaves. That gets a little bit complicated because sometimes those plants do not have the exact same genetic background, and you don't know how much they've changed. So doing things in the lab with bacteria, it's really a lot more fun and easy. So we use the strains from Cuatro Ciénegas to explore this phenotypic plasticity across and within these genotypes, and with strains that have been exposed to the same environmental conditions. So if we think that these bacteria in the Turince pond have been there for hundreds and hundreds of years, and they've been all subject to the same temperature changes, there's no reason that we could think that some bacteria are going to be more tolerant to a shift in temperature than the other one, because they've all been selected for hundreds of years to live in the same temperature. However, if we actually take two different groups of bacteria, all from the same genus, which is Bacillus, and then we test different strains of Bacillus cereus, different strains of Bacillus subtilis, and then we look for their reaction norms to changes at different degrees of temperature and see how well they grow. What we see is that those bacillus that belong to the B series group, then they have different optimum growth phenotypes at the different temperatures. And those that belong to the B subtilis, they also have their own plasticity. And so this is interesting because they were all grown for centuries in the same environment. So we think that it's really their genetics that is conditioning the plasticity allowed for these strains. So how about the genotypic plasticity? In bacteria, there's a lot of exchange. We already heard Esperanza, and we see that things change a lot in bacteria. They lose genes, they gain genes. There's a lot also of plasmids and prophages, transposons. So there is usually a thing that the bacteria have like a core genome, and then they have a sort of like a genome that can be really, changing depending on the environment. So it is typically that if we take 3 strains within a given genus or even within a given species, and we look at their genomes by using complete genome sequencing, what we will typically see is that by the 16S gene, which would be a single highly conserved gene, we could deduce and we could decide that bacillus A, B, and C are almost identical, they can be the same species. But then, if we take a look at what's going on in the rest of the genome, what we will find is that there can be 30% difference in the genes that are present there. And these differences in these genes, in fact, are going to be those that we call accessory genes, that are those that define the niche of the bacteria. Those have the ecological traits, things that allow it to scavenge for phosphorus or for the different carbon sources. So the bacteria that is in our model has this property that it can form spores. The spores, look like in phase contrast. You can see them as these, phase bright, little round things within the bacteria, and these are, thermoresistant. But these bacteria also can form biofilms, and they also make flagella, so they can swim. But they can also have what we call social traits. Under different conditions, they can all move together. They can all make flagella and then produce surfacting and then move in this social movement. So both biofilm and swarming are considered social traits. So how plastic would you think bacteria are in this genus to sporulation? So sporulation is the defining trait for the bacillus. And yet, if you look at what's going on with the genes that are known to be involved in sporulation, and that comes from the Bacillus solaris model. So the very left column, all in gray, in light gray, it's all the genes that are needed in Bacillus solaris to sporulate and to be competent. Competent is the ability to take DNA from the environment. So these are all the genes that we know were important for sporulation. But if we look for those genes now in other bacillus that we know also sporulate, what we find is that half of the genes are not present in other bacillus. So they all sporulate, and yet sporulation genes are not part of the core gene. They really change a lot, and it really is telling us that there are many ways to form a spore, and that even this trait is highly plastic. So I'll tell you a little bit about the microevolution of a particular bacillus, one that we've called Bacillus coahuilensis. And Bacillus coahuilensis, we've sequenced 3 strains. We thought that because they were the smallest genomes of the bacillus, and because they lived in this oligotrophic place, it was like, how much can they evolve? How different can they be? They are already constrained by the environment and by the genome size. And what we found is that they are just as dissimilar as any 2 E. coli's or any 2 things that you sequence in other places. You have about 30% of the genes that are different. And 6% of the genes belong to a category of these mobile elements. You can have phages, you can have transposons. So that's, again, the plasticity of these bacteria that rely on these genetics and having all these mobile elements to be able to change. So we did this experimental evolution with Bacillus coahuilensis. Again, we thought, okay, this is a bacteria that doesn't have a lot to, a lot of ways to evolve. It's already a small genome. So we took just one of the strains, which is strain 26, And we made 6 populations, and we just took it in minimal medium using glucose as a carbon source. And after just 100 days, we said, okay, that's enough. Let's see if we can find difference here. And we then took these populations and analyzed the capacity to use different carbon sources. So it's evolved under glucose, and then we take 100 different carbon sources, and we determine if it can use those things. And I'm not going to show you the 100, but all the graphs look the same. They're all identical. And what happens is, if you have the ancestral always in black, so the ancestral is strain to 6, and after 100 days, you have these 6 populations. So you have the red, the green, the blue, etc. And you see that under glucose, they all not just can still use glucose, that's how they were evolved, but they are a lot more robust and efficient. They are much better at using this substrate. But then they can also use better maltose, maltodextrose, sucrose, trehalose, and lots of things. So what happens is that, it doesn't matter that you just evolve them under a single, source. They can really use, many more. And we also looked at, whether the mobile elements were really moving, or are these things alive? We can still, shift these transposons, and we found, yes, we can find evidence that some of the transposons are moving. So there is some genetic plasticity here. And basically what we conclude is that we have this phenotypic optimization after growing them in glucose. And basically, the only way to explain that glucose optimization can select for optimization of another 30 different carbon sources means that the optimization is really not, it's being driven by a particular source of carbon, but it's really a substrate on specific adaptation. So really, we think it's regulation, what we're seeing, and sort of like a little switch in regulation can have this big effect on the phenotypic ability to use a lot of things. So basically, in the pond, Bacillus is not going like it's to its full, optimum for usage of different sugars. It's actually pulling it down to the minimum, that it's needed in the pond to live. But it really has the potential to just amplify this repertoire of carbon sources that it can use. And now we are in the middle of sequencing this and see that, that is going to be probably a regulator. Okay, now, another thing that we looked at was, other traits, like the ones that I showed you. I showed you that bacillus could swarm, and could also make biofilms. But can all these bacillus in the communities, really have these traits? So, well, one of the things that we started with was this prototrophy. I mean, bacteria are known to have all the pathways to synthesize all 20 amino acids. And that's what you would expect, of a robust bacteria, that it can make all its amino acids. And that is the case of Bacillus subtilis in this clade here. So these blue little circles mean that this bacteria is prototroph. It's able to make all of its amino acids. And when you see no blue thing, or it's little, just light gray, it means that it's an auxotroph. So most of the bacteria that we isolate from communities are actually auxotrophs. They really rely on the community to be able to get the food, especially their amino acids. So in some of these, for instance, in Bacillus coagulensis, what we've noticed is that for the auxotrophy for valine, isoleucine, and leucine, the phenotype is like you have to add these to the medium. If you look at the genotype, you see that the pathway for the synthesis of valine, isoleucine, and leucine is eroded, so they lack a lot of the genes involved in the synthesis of these. However, if we look at valine and alanine, it just are not able to use these they really need these amino acids in the medium. But if you look at the genome, you see that the whole pathway is intact. So we have no idea what's going on there. Okay, so then we studied another group of Bacillus cereus, and we noticed that these were 18 strains, and they were all auxotrophs. They all needed leucine and valine, or only valine. And what happens is that when we sequence them, because I told my student, Diana, I said, Diana, this is great, now we have a system to look how these pathways are eroding. So no eroding, nothing, it's all positive, the pathways are complete. There's, all these 18 genomes, we couldn't find any evidence of something going on. So what we think here is that, again, there's a regulatory switch, that it's really just, not allowing them to, they're responding to something, because if we put them together, cells, like this strain will not grow, by itself it won't grow, but if you put them in this mirror, so that's just what you expect in a community. You expect in a community, things are being shared, and bacteria are either killing each other or helping each other, but really this community is really the basics that you have to have plasticity, because you don't know who is going to be your neighbor tomorrow, and you have to be ready to change and evolve, or to move someplace else, or do something. So now, after prototrophy, we also looked at the social, phenotypes. So can these bacteria, swim? Can they swarm? Can they form biofilm? So let's go from the part, at the outside circle, it's swimming. Most bacteria are able to swim. Then we go in the middle ones, and this is the yellow one, or something kind of yellow, it's the swarming. Again, Bacillus sololis can swim, can swarm. In fact, it can make biofilm, it can do everything. But these traits are lost in many of the other, taxonomy groups. So the question is, are all these genes lost? I mean, am I going to get another, student and sequence more and find that things, flagella are there? But I bet it's going to be there. Well, we are in fact sequencing everything, yes, but this time, I'm not betting that these pathways are being eroded, and my bet is that these pathways are all responding to their neighbors, and that there's a lot of complementation in molecules going on, and a lot of signaling. If we look at the presence of one of the enzyme, particular enzyme that is important to gather phosphorus from the medium, we see that Bacillus coagulensis, two of the strains have the alkaline phosphatase gene. However, if we look at the phenotype, we see that two of the strains will respond, will produce the alkaline phosphatase if there is no phosphorus in the media. But with phosphorus in the media, this one is going to shut off. That's what you expect, regulation. So this would be constitutive, these will have no alkaline phosphatase, and these will be regulated. So you have the same species, 3 strategies. So basically, again, you have all this plasticity that is allowing to conserve genes or lose genes, and everything is possible. So this is explained in ecology by a term that is called compensated trait loss. What this means is that in nature, a given bacterium can lose genes if the product can be obtained from the community. A particular application of this compensated trait loss is black queen effect, in which bacteria are just not going to be collecting genes because it's going to be energetically expensive. So if there's someone else in the community doing something, they will prefer to lose the gene and let the neighbor, give them sugar or a tomato or something else. So that's one way to see this. But then another way to see this is that gene regulation is really, one of the important things in heterogeneity that you observe in these communities. And this, I love this phrase by Costerton. He is an expert in biofilm, and says that although the bacterial genotype is extensive, the ability of the genotype to respond phenotypically to environmental stimuli rather than the genetic repertoire is what gives the remarkable success of bacteria. And so I think this is important. Again, this is the interplay between the genotype and the phenotype. And how am I doing with time? Am I? Yes? Okay, so then I'll just tell you, I told you that in Quadracenigas, phosphorus is highly limited. So how are these bacteria recycling phosphorus? In the ocean, this is a very important thing because the phosphorus, is in different, biochemical or chemical states. And you have the cyanobacteria recycling it. So what happens in Quadracenigas? So I'll show you that one of the things we looked at was first how much phosphorus do we have available in the soil or in the sediment. So the comparison is going to be soil versus sediment. And we find that dissolved phosphorus in the soil is pretty low, but it's pretty high in sediment. So it's like, okay, so wasn't it just limited? I mean, so what's happening here? It's not that limited at all in the sediment. So if you look at the enzyme expression in sediment and in soil, what you find is that these monoesterases and diesterases, they are highly expressed in the case of sediment. So I think this is a little bit of where we want to go with our work, is in trying to see how the enzymatic activity is really changing the environment. These enzymes that are made by the bacteria are actually releasing phosphorus in the community. So you find this free phosphorus for the full community in the soil. And then that's what we call that it's public goods, because now bacteria that can dissolve some form of phosphorus are getting this available phosphorus that now for the full community. Okay, they have no way to just have the phosphorus for themselves. So, this is, again, now I think that we find this evidence that we are seeing is that how bacteria are producing enzymes, and these enzymes are releasing phosphorus. It's a little bit, brings us a little bit to understand how this is going on in these communities. So we looked at different substrates that bacteria might like to eat, and we looked at the preference of 1,000 of bacteria and of all bacillus, and see what is the heterogeneity of these bacillus to use phosphorus. And I know that I hate these myself, okay? I'll go really, with you, and I'll show you what this is. So you have here the typical, phylogeny of the bacillus. You have a lot of bacillus organized here by their historical evolution. And then what you have here is that the black bars indicate that these bacteria are capable of using the substrate. And the substrates are the ones here. We have on the outside DNA. So DNA has a lot of phosphorus and nitrogen. And bacteria love DNA as a source of phosphorus. They like it even more than if you have that as a form of potassium phosphate. So all of these black things mean that all the bacteria can use DNA. And then you have phosphonates, and phosphonates, some use it, but it's not really an exciting source of phosphorus. And then you have phosphate, which is another chemical form. And I'll just, well, you get the idea that phosphorus utilization capabilities are distributed. It's not by a single taxon. Okay? So there is one particular group there that is the Bacillus cereus. And although these are genetically very, very similar, those that are in soil and those that are in sediment have different abilities to utilize phosphate. So those in soil, only 12% of those can use phosphate, while 70% can use it in sediment. So, again, this ability to use phosphate depends on a single gene. In fact, it's two genes. So these two genes are easily moving, bilateral transfer, and they are being selected, in bacteria that live in sediment. Okay. So in phosphorus, what we see is that phosphorus, is distributed. So not all strains have to do everything. It's a little bit like the black queen hypothesis. Get rid of some genes, let your neighbor, do the job of liberating that phosphorus, and we can all use it, and we can all save, resources. And these enzymes then are public goods because they can be, influencing the neighbors and allowing all to have the free phosphorus. And just to conclude here, and then warning, the more you look into what the species concept is and you look at the metagenomes, the more careful you have to be about the ecological predictions. Just looking at the 16S is not telling you what's the preferred source of phosphorus. It's not telling you what the preferred, source of carbon sources. It's not even telling you if it's going to be able to swim or swarm or make a biofuel. So there are a lot of things that you think you know once you know the 16S and the identity, but you really don't. And then the other thing is that the different taxonomic groups are different. The Bacillus solaris group is very robust, and the other ones, seem to be really feeble and they lose everything. And in these communities, then maybe things are just not mixing. Maybe they all are doing their own business. They have a structure, and biofilm and spore are barriers to exchange of public goods. So maybe that's something we are exploring. And the phenotypic plasticity can occur at micrometric distances. I mean, we don't have to think that this is happening kilometers away, because even in a small community, if you are making an antibiotic, that antibiotic at some distance can have a dose that will kill the neighbor, but if it's a few micrometers away, it can be a signal to swim away. So, this is what's happening, at small scale, and that's also something, to think about, that the concentrations of metabolites in communities can change very rapidly. So, again, I think that plasticity of the genomes, it's an adapted, trait. Bacillus solaris don't seem to be able to change that much. Even when we do look at the reaction norms, we find that Bacillus solaris, that cannot change anything and never loses genes, is the one that has, more plasticity. And we have the Bacillus cereus that has larger genome or larger repertoire. It's more constrained in the things it can do. And that's something that has been suggested to occur in plants. So species is a real concept in bacteria, but we think that the taxonomic groups are hardwired. There is plasticity, but there are boundaries, especially in the size of the genomes of the given species. We don't find, even in Patrocienegas, where we thought that this environmental constraint was going to make smaller genomes, we find that each genome really has the same, size as if it lived in any other place. And with this, I'll just say thank you to a lot of excellent collaborators, and of course, thank you to CONACYT for the money to do a lot of things, and have excellent students. They have posters, and then also people from UNAM and UNAM CIECO and Inés Morelia that are great collaborators too. In Monterrey, in CIMBESTAF, I also collaborate with Moises, and with Mike Travisano and Gabriel Moreno and Jim and Susana in Nuevo León. And thank you very much.